All right, this is part 2 of review of renal physiology, and just to pick up where we left off, remember, so far, we went through basic kidney anatomy, and we've talked about two major things that involve regulating both GFR and blood pressure / blood volume. And the first part of that was autoregulation. Remember, autoregulation is just the kidney taking care of itself. This is the kidney effectively saying, I want to keep GFR relatively stable at homeostasis. If blood pressure goes up and down, GFR would go up and down. And so all this is really saying is if blood pressure goes down for some reason, the arterial, right, that afferent arterial going into the glomerulus has to dilate to let more blood in to get your GFR back up, or it would go down. If blood pressure went up, you would vasoconstrict so that you don't overpressurize and have GFR go up too high. Right? That's autoregulation. And the second half of this tubuloglomerular mechanism, I'll get to before too long. The second part of it is the hormonal aspect of controlling GFR. And I guess what I want to make really clear with this is we're talking about kidneys, and so I have this slide titled Regulation of GFR. But the primary purpose of the renin angiotensin system is really to control blood volume, and blood volume affects blood pressure, and blood pressure affects GFR. And so here, the primary concern is blood volume and blood pressure, and GFR is just sort of secondary. Okay, so remember angiotensin and aldosterone, this is your response to low blood pressure systemically, and ANP is the response to high blood pressure because of high blood volume systemically. Okay, they do affect GFR, but again, their main concern is volume and pressure, whereas the autoregulation stuff, that's just the kidneys, taking care of its internal situation. Okay, these are systemic. So the place we're going next is describing what's actually happening in the specific segments of the renal tubule itself. And the gist here, so this is showing you a glomerulus up top. This would be the start of the renal tubule, so this would be PCT. And they don't draw it in the rest of these slides, but there are capillaries that run all up and down the renal tubule. So PCT, the loop, the DCT, the whole thing, there are blood vessels all over the place. All that's really saying is when fluid enters the renal tubule and becomes filtrate, right? So remember that number that we've been throwing around, that average of 180 liters a day, if you added up all the nephrons in both kidneys in a normal-sized person, you don't form anywhere near that much urine. And so all this is really saying is the vast majority of that water is going to have to come back out and be reabsorbed. And when you talk about, reabsorption, that really is just saying the bulk of that fluid gets taken out of the renal tubule, whatever segment it happens to be in, and gets put back into the blood. And there might be a little bit of secretion back into the tube too. Don't really worry about that. The two big players are, what's your GFR? That's up here. And then how much do you reabsorb down here? And so in our ballpark, if that's 180 and a normal urine volume is somewhere around 3, then this would be our 177 left over, right? The vast majority of fluid going into filtrate gets put back into the blood, right? That's the general concept. The details are what we're going into next for each particular segment of the loop, and there's going to be a little bit of jumping around over the next 4 or 5 slides, but I'll try to make it coherent. So, PCT, this is pretty straightforward. This is where the majority of fluid gets reabsorbed, so about 2/3. That means that if you put 180 liters in at the glomeruli into GFR, only about 60 of that makes it down into the loop, and the other 120, right, 2/3 of 180, must have been reabsorbed into the blood out of the PCT. There are a few things going on here. One is, obviously, that's a lot of water. The primary purpose is if you are taking all of your waste products and toxins and things like that and letting them into the filtrate, if water gets reabsorbed, those toxins and metabolic wastes are being trapped inside what's left. And so if the amount of water is going down, the concentration of those waste products is going up, which is obviously what you want because you want to end up with urine that is concentrated in whatever you want to get rid of. The other thing that's happening here is try to get some detail on this. These guys, so glucose, amino acids, they say other nutrients. Remember, these are things with calories. So that list I made in the first video, caloric nutrients, you're going to take back as close as possible to 100% of those nutrients in the PCT. That's where all the microvilli and mitochondria come from. If you would look at this really close under a microscope, I don't have an EM, an electron micrograph of this to see those, but it's very similar to the small intestine. Right? Remember, you can't stop glucose and things like that from getting into the filtrate, and you want them back. Because if that isn't working properly, that's what happens in an uncontrolled diabetic. If their blood sugar gets so high that the active transport pumps in the PCT can't get all the glucose back in quickly enough, they'll wind up with glucose in the urine, which is harmful for a lot of reasons. So hopefully you take all of that back. Okay? These things that are non-caloric nutrients, sodium, bicarb, that's a really long list, potassium, chloride, magnesium, on and on and on. We'll say most of that gets reabsorbed here, but not all, because those are things that need to be fine-tuned. Right? Glucose, you take it all back, because if you have extra, you just store it. You can't store sodium and bicarb, and so that's going to be some fine-tuning later on over in the DCT to control the precise amount that's in the urine and the precise amount that's back in blood. Okay? So this is mostly take the bulk of your water back to concentrate wastes, and then, at the same time, make sure that you get nutrients back that have calories in so they can be stored somewhere. All right. The descending limb. This is where I'm going to do things slightly out of order. I'm going to jump to slide 24 for a minute and show you, this is a really, really important slide for showing you what's going on with the renal loop and the collecting duct. Okay, all of this ties into the hormone ADH, or antidiuretic hormone. What's going on here is you're fine-tuning exactly what the concentration of your urine is. And remember, concentrations have two parts. This is considering how much water you have and also how much dissolved stuff you have. It's easy to say salts, but it's not technically all salt. A lot of it is sodium, but there are other minerals. There's urea, there's uric acid, there's metabolic wastes. This term osmolality, right, which is also the scale, these numbers that you see written all over, those are milliosmoles. And you can kind of think of that as saltiness, but again, like I said, it's not all salts. It's if you take up everything dissolved in a liquid and add it all up, basically counting the number of molecules, that's osmolality. Okay? And so if you're dehydrated, the definition of dehydration is having a high osmolality, right? And you think of having, being dehydrated as not having enough water, and technically that's true, because if you wanted to decrease osmolality, you would just add water, okay? But this is, again, where you have to very clearly separate concentration, that's osmolality, from volume, right? Aldosterone, angiotensin, ANP, those are volume issues. Just how many milliliters of water do I have? This is a concentration thing, right? What's my osmolality? So, for right now, the gist is, this is showing you a schematic of the loop and the collecting duct in the kidney. Everything from here up is cortex. Everything from here down is medulla. The shading on here is showing you that as you go deeper down into the medulla, the osmolality of the fluid in the kidney goes higher. It gets saltier, right? And so you start off up here in the cortex with an osmolality of 300, and by the time you get way down into the medulla, like at the base of one of those renal pyramids, it's increased about 4 times. So you're up to around 1200. And the reason for this is there's no such thing as a water pump, right? So there's no active transport pump that moves water, but we have to be able to move water, because if you're dehydrated, right, like you're up here, you release ADH. The goal is to retain as much water as possible. You want to take that water out of the collecting duct so that it doesn't end up as urine. That's why you get a small volume of concentrated urine at the end. All right, so we need a reason for water to be sucked out of the collecting duct and put back into the blood. Right? They're not showing capillaries over here, but they're there. Right? If you can't pump water, you can pump salt. Okay? And so the whole point of this renal loop is, the most important part of it is these red arrows. If you look at the key, red arrows are active transport, blue arrows are facilitated diffusion. or passive transport, the purpose of pumping that sodium is to build that gradient, right? To make the kidney salty down in the medulla. And the reason is water follows salts, okay? If you want water to move, use these little diffusion channels called aquaporins. Not active transport, just regular diffusion. The catch is water follows salts. So if you make the kidney really, really salty, right, water will effectively chase after those salts moving through aquaporins to try to reach equilibrium. It'll never happen because this loop is constantly pumping in sodium to make sure that the salt gradient doesn't go away. And so all that's really changing here is if you look at the two sides, what's going on in the loop on both sides? Exactly the same. The only thing that we're changing is if you're dehydrated, ADH's target cells are in the collecting duct. The thing that it does is add aquaporins. Aquaporins are water channels, and so you get that water reabsorption back into the blood, small volume of urine. If you're overhydrated, meaning your osmolality is low, you want to get rid of water. Right? ADH levels go down, aquaporins go away, and all of that water that's in the collecting duct stays trapped in the collecting duct and goes out the kidney to urine formation. That's why you get a large volume of dilute urine. The water is not going back into the blood because there are no aquaporins there, and that's exactly what you want because you're overhydrated. Okay, so the whole purpose of this setup is control the concentration of urine is the biggest part, okay, depending on whether you're overhydrated or dehydrated to start. And the really, really, really important thing for this exam is, and I've said it multiple times, but it's very hard to wrap your head around sometimes, is volume and hydration are completely separate things. So if on the exam, if I ask a question about hydration, you really only care about ADH. That's it. If I ask a question about volume, you care about aldosterone, angiotensin, and ANP. Those are your volume hormones. And one doesn't predict the other. So if I would ask you on the question, or on a test question, ADH levels are very high, that would mean a person is dehydrated. What do aldosterone levels look like? You would say, I have no idea, because ADH is the response to hydration, and aldosterone is a response to volume, and the two don't really connect directly to each other. They do indirectly, and so all that means is you can be dehydrated with a high blood volume or dehydrated with a low blood volume, because again, hydration is simply osmolality. How salty is your blood? Okay, so the details about that loop. The descending limb is just more water removal. This is more concentrating of waste products, and so water diffuses out, chases that salt gradient that we already talked about. That's pretty straightforward. So there are aquaporins there, but they don't ever change. They're always present. They don't respond up and down like the aquaporins in the collecting duct do to ADH. The ascending limb, again, active transport of salts, primarily sodium chloride. That's the big driver of building the salt gradient. We already covered that, so there's not really a whole lot else to do here. Okay. The DCT is where fine-tuning of non-caloric nutrients happens for the most part, right? And I'm only going to use a couple examples. One of them you really already know. So this is the target site for aldosterone. Aldosterone turns on active transport pumps that pump sodium back into the blood. Since water follows salt, that increases blood volume. That's this, right, from aldosterone. And I already, when we first went through it, mentioned that happens in the DCT. Okay, so that's the same thing. It's just giving you, a bit more context as far as where it's happening in the renal loop. Okay. The second example I'm using that you also already know from the endocrine section, right, when we went through this back at the beginning of the semester, is this is also where target cells for PTH, parathyroid hormone, are for calcium homeostasis. And so if someone's low on calcium in their blood, remember PTH gets secreted from the parathyroid glands, targets the epithelial cells of the DCT, and causes calcium reabsorption. Because if your blood calcium levels are low, you don't want any calcium winding up in the urine, because that'll just make the problem worse. Right? And there are other examples of this for other minerals as well, but these are the examples that we're using for now. Okay, and then again, lastly, the collecting duct, again, I've mostly been through this already, but just to reiterate, this really is the target site for regulation by ADH. Entirely hydration, nothing to do with volume, is the major determinant of urine concentration. And again, its only real effect is adding or subtracting aquaporins. The salt gradient's always there. ADH just controls whether the aquaporins are there to let water actually move or not. Okay? And I think the next thing I want to do is to illustrate the limitations of this. This max number around 1200, that's as salty as your kidney gets. So if you eat or drink something that's saltier than that, it's not going to help this problem. It's actually going to make it worse. So as an example, if you drink ocean water, which it's probably hard to do without vomiting, ocean water has, I think, someone can fact check me, a osmolality of somewhere around 2000, maybe slightly higher. If you drink that, you can't form urine that's 2000 milliosmoles. You have to dilute it down to 1200. The only way to do that is to add water, and the only place that water can come from is from your blood and from the tissues in your body. And so the reason why drinking really salty water or eating very salty food makes you even more dehydrated is you're forced to steal water from the rest of your body just to be able to form urine, because that's your max concentration. Right, this is why salty food makes you thirsty. It's the same reason why when restaurants and bars give away free snack food at the bar, it's usually salty stuff because it makes you want to drink more. That's part of how they try to increase the profits. On the overhydrated side, the lowest you can get is around 100. You can't form urine at 0. That's why if you keep drinking more and more and more and more water, eventually you'll start losing even more salts and become severely overhydrated, and then you'll get low levels of sodium, hyponatremia, and that in itself can cause huge problems because all those ions are a huge part of controlling neural signaling. Okay, so as long as you stay between these two numbers, the combination of whatever you're eating and drinking is somewhere in that range, you're good. If you go too high or too low, you're basically outside of the kidney's range of abilities as far as what it can regulate. Okay. So there's a little bit more to do here, but this stuff that I've gone over so far really is the most important content for this exam. Put a lot of focus into regulatory mechanisms for hormones, aldosterone, ANP, ADH. I guarantee you there'll be questions asking about all those hormones, what their effect is, where their target cells are, what problem are they fixing. They're all negative feedback loops. So try to structure, your studying in that way. And we can probably talk more about that on the, review sessions at some point. Okay. All right. I guess one thing I have to go back and fill in before I keep going. Remember, we skipped over part of autoregulation, and before I jump back to that, I want to remind you that the ascending limb of the loop, its primary job is to actively transport sodium chloride. Those pumps, those active transport pumps that are moving salt, they have to have time to pump that salt. And if GFR is too high and this fluid is racing through too fast, those pumps won't have enough time to remove the sodium, and sodium levels up here will be too high. And if the fluid moves too slowly, there will be too much time, the pumps will take out too much sodium, and sodium levels up here will be too low. That is effectively what's happening with that tubuloglomerular mechanism. You're sensing sodium chloride levels in the ascending limb of the loop where I just was. And if, again, in their example, GFR is down, flow rate is down, that means that there's going to be effectively too much active transport. And if that's the case, you need to vasodilate to get GFR back up to get fluid moving again. And on the flip side, if pressure was high, GFR would be high, which means sodium levels would be high. You reverse everything and you'd wind up vasoconstricting, right? And anatomically, that's all happening back here. This is, I know it's kind of strange, this is the PCT coming off the glomerulus. This is the very top of the ascending limb of the loop. So the loop is basically behind the screen, coming up out of the screen at us. and those maculadenza cells are sensing sodium chloride levels and then releasing chemicals like nitric oxide that will vasoconstrict or vasodilate the afferent arteriole that's needed. All right. Okay, well, the rest of this really is just adding a few details and cleaning up a few loose ends. This is a minor point. All along, we've been acting like all nephrons are the same, and this is just effectively it's really just a caveat saying there are two different kinds of nephrons. Cortical nephrons that live in the cortex and have big PCTs and DCTs and pretty much do all the functions associated with those segments. Juxtamedullary nephrons, because they're juxtaposed to the medulla, they have really big renal loops, and they pretty much do all the stuff with making the salt gradient. And so there's nothing really new to know here, except there are actually two categories of nephrons, and we've been sort of lumping them together all along. But for the sake of the exam, you don't really have to worry about this separation too much. All right. Urea, again, this is more just general awareness. Urea is a very important waste product. It's pretty much always found in urine, and it is the waste product of amino acid metabolism. So when you eat protein, amino acids have nitrogen in them. If you think about the cellular respiration equation, the end product is CO2 and water. There's no nitrogen there. And so you have to have a way to get rid of the nitrogen from protein. Like if you're using protein to make ATP, that somehow is urea. You would initially form ammonia, but that's incredibly toxic. And so it gets converted into urea, which is very non-toxic. And part of its role in the kidney is urea is part of what makes the salt gradient. And the reason that it's important is because if you did the whole thing with sodium, it would be toxic. That much sodium would mess with cellular function. And so this osmolality gradient is a mixture of sodium chloride and quite a bit of urea as well. Okay, pH. Kidney also has a really important role in pH regulation. It's not buffering, right? It's not a buffer like the respiratory stuff that we did for the last exam. Because remember, buffers really just hide protons. They, grab onto them and let them go depending on whether you're acidic or basic, but they can't make them actually go away. You can breathe them out as CO2, I suppose. The kidney, on the other hand, is not as fast as a buffer. It's not an instant, chemical reaction, but the kidney can physically get rid of protons. And since humans pretty much always lean towards being acidic, like if you just let a person go, since you're always making CO2, you trend towards acidosis if it's uncontrolled. And so the PCT has a mechanism to actively transport protons. And you don't have to know details from this, but the gist is you're doing active transport of protons as needed. So if your blood starts becoming a bit acidic, which it pretty much always will just by default, anytime you eat, you're pumping those protons into the urine. That's why urine usually has an acidic pH. Okay, so for the exam, I may ask you to do a compare and contrast between how the kidneys regulate pH versus how blood buffers do. And the short story is blood buffers are very, very fast, but they're limited because they can only hide protons. They can't make them go away. The kidney is slower, but it's a long-term solution that's permanent because it can physically remove the proton from your blood and actually get rid of it, right? Flush it out in the urine, and it's completely gone. It's not a buffer. It's a removal system. Okay. This, I would like you to have the general concept of this. I'm not going to make you do it in detail for the exam. Measuring renal clearance, usually the initial red flags for a kidney problem is having high blood levels of waste products or minerals. Urea, creatinine, or creatinine, some people say, or uric acid. If you have high blood levels of those things across the board, it's a pretty good sign that kidney function is down. And if your GFR keeps decreasing, it's just saying that you can't clear waste products fast enough and they start to accumulate. Right? If you really want to actually carefully measure GFR, the closest you can really get to it is a renal clearance test, and to make that work, you have to inject a person with a molecule that only the kidney can clear. it can't be metabolized by the liver or any other organ. The current sort of favorite molecule is a plant carbohydrate called inulin. You inject it into a person, it's passively filtered out their kidney, and effectively the higher their GFR is, the faster it goes away. And so if I inject somebody with inulin, we'll say that that's inulin concentration, and they were at 0, and then I give them an injection so their blood level spikes, and I measure it over time, you would expect to see something like this on a half-life scale. And if that's in blood, if you would measure inulin in the urine, let's say that's the red line, you would expect to see a mirror image like that, because if it's leaving the blood through the kidney, the only place it can wind up is in the urine. And the x-axis in all of this would be time. So you're really asking the question, how fast does this happen? And the faster the inulin's out of the blood and into the urine, the higher the person's GFR is. Okay. If it gets bad enough and the kidneys just can't do their job, this is usually the result of diabetes or chronic hypertension that damages blood vessels, then really the only options are dialysis or transplant. And a lot of people wind up on dialysis because there just aren't enough healthy transplantable kidneys to go around. Okay. All right, and last thing, kidney stones. This is more, I put this in my lecture notes because usually someone ends up asking about it. These are chunks of crystallized minerals. Calcium is a big one, but others as well. And they form in the renal pelvis for the most part, but don't really cause pain until they're in the ureter. So, in this illustration, if you imagine a kidney stone forming somewhere down here, doesn't really cause any pain or problems, and then all it takes is the right movement. Oftentimes it happens at night when you're laying down, to lodge inside the ureter. Remember, the ureter has lots of smooth muscle around it, and so as soon as it relaxes to let urine go through, the stone slips down into the ureter. All that muscle will then contract to prevent backflow, and the muscle effectively clamps down on the stone. And the preferred method to get rid of them now is basically blasting them apart with high-intensity shock waves, which is better than surgery, but is still very painful because the fragments still have to be passed at that point. Okay, so that's kidneys, and I want you guys to send me questions. We're going to have review sessions before the exam, so sending me those questions ahead of time is helpful. Then I can prepare for answering them more efficiently.